Good afternoon, everybody, and welcome to today's Global Health Compound Design webinar. This is the fourth in our recent series, which was set up to support global health scientists working in these uncertain times. We set them up as we all face our own global health event in the form of the COVID-19 virus, and I believe we have a number of AbbVie's virtual interns listening in today. So I'd like to point them towards MMV's website, where there's a listing of all the events that are going on, and also links to all the recordings, which you can find under the you have to know where to look up for this. You have to look under the computational chemistry page, and these meetings have been going on since 2016, so there are lots of resources there that you can use and listen to learn more about drug discovery. Right, whilst I have your attention, allow me to announce the next session, which will be on Thursday, the 25th of June, and will feature Neil Berry from the University of Liverpool. And can I also encourage anybody who wants to ask a question to type it into the chat box, and we'll put them to Tom at the end of his talk. So it's my pleasure to introduce today's speaker, who is Tom von Geldern. Tom trained as a chemist at MIT and the University of California, Berkeley, before joining Abbott Laboratories, where he spent the majority of his career, most recently serving as a research fellow and senior group leader. And in 2007, he left to become a consultant specializing in medicinal chemistry and discovery strategy and tactics. He's maintained his association with AbbVIe, has worked with DNDi and a range of other global health organizations. He is a true local hero, and in 2017, he was named as Chicagoan of the Year with Howard Morton for his work on, in fact, on this project. And it's my pleasure to invite him to give his talk on Tylosin analogues as antifilarials. Okay, good afternoon, everybody, and welcome to today's Global Health Compound Design webinar. This is the fourth in our recent series, which was set up to support global health scientists working in these uncertain times. We set them up as we all face our own global health event in the form of the COVID-19 virus, and I believe we have a number of AbbVie's virtual interns listening in today. So I'd like to point them towards MMV's website, where there's a listing of all the events that are going on, and also links to all the recordings, which you can find under the you have to know where to look up for this. You have to look under the computational chemistry page, and these meetings have been going on since 2016, so there are lots of resources there that you can use and listen to learn more about drug discovery. Right, whilst I have your attention, allow me to announce the next session, which will be on Thursday, the 25th of June, and will feature Neil Berry from the University of Liverpool. And can I also encourage anybody who wants to ask a question to type it into the chat box, and we'll put them to Tom at the end of his talk. So it's my pleasure to introduce today's speaker, who is Tom von Geldern. Tom trained as a chemist at MIT and the University of California, Berkeley, before joining Abbott Laboratories, where he spent the majority of his career, most recently serving as a research fellow and senior group leader. And in 2007, he left to become a consultant specializing in medicinal chemistry and discovery strategy and tactics. He's maintained his association with AbbVIe, has worked with DNDi and a range of other global health organizations. He is a true local hero, and in 2017, he was named as Chicagoan of the Year with Howard Morton for his work on, in fact, on this project. And it's my pleasure to invite him to give his talk on Tylosin analogues as antifilarials.
So thanks, Caroline, for the nice introduction, and welcome everybody. Good morning, good afternoon, good evening, depending on where you happen to be sitting. It is definitely a pleasure for me to be able to share the results of our team's research into the development of potential new treatments for filarial disease. Before I start on the science, I do want to put up what is the most important slide in my deck, actually, which is the list of the folks who have participated. You can see it's quite a list at this point. In fact, this project started out very small, but as these things happen, as it moves forward and as we begin to have some success, lots and lots of people have joined in, contributed their skills and their energy to help it move forward at various times, and we absolutely appreciate that. I want to highlight the worm experts, the folks from Joe Turner and Mark Taylor's lab at the Liverpool School of Tropical Medicine, Akam Hurov, Mark Hübner and their colleagues at the University Hospital in Bonn, and Sam Wanji and his team at the Research Institute for Tropical Diseases in Buea, Cameroon. These folks taught us a ton about worms over the course of this project, and they are the ones who've been responsible for doing the in vitro and in vivo profiling of our compounds. Need to thank the filarial team at DNDi, Ivan Skandali, Sabina Specht, and Rob Don and others who've been very supportive throughout this project, and actually whose role will continue to expand as the program moves forward. And then absolutely I need to thank my friends and colleagues at AbbVIe. Just a reminder, these are all folks who have full-time jobs developing medicines that the company can actually sell to make money. And so, but they have enthusiastically embraced this program and have volunteered their time, their energy, and their effort to do critical experiments and to help us move forward at key moments. Caroline mentioned Howard Morton. I want to highlight him. Howard was having a perfectly happy time in retirement from Abbott when he was dragged back. He did all of the process chemistry work on this project, developed the route that now has been used to make multi-kilo quantities of our clinical candidate material that has been used for not only for preclinical studies, but also to do the clinical trials that are underway. I'll mention Kenan Marsh, for those who know her, a force of nature. Kenan has coordinated all of the preclinical pharmacokinetic and safety profiling work for the program. And then also just want to mention Dale Kempf, the head of neglected disease research at AbbVie, and also Jeff Rohde, a former Abbott scientist, now professor of chemistry at Franciscan University. The two of them developed the strategy that allowed me to be able to get access to space in a chemistry lab at AbbVie and also access to infrastructural support to be able to work on this project. You might think that it would be easy to volunteer your time for a major corporation. I assure you that that is not the case. And so the two of them were very resourceful in allowing that to happen and allowing me to be involved, and I appreciate it. So let's talk about our diseases. These are diseases caused by infection with filarial or thread-like worms. The first that we'll mention is onchocerciasis. Worms of the genus Onchocerca are spread typically by biting flies. When those worms are in their human hosts, they then grow and develop. The females, who can be quite large, actually begin to build nests, and then they go forward and produce juvenile worms of their own, what we call macrofilaria. These macrofilaria can migrate to the skin, where they cause severe itching. That's one symptom of this disease. And they can migrate to the eyes. And there the body's inflammatory response to their presence can lead eventually to permanent clouding of the lens, hence the more common name for this disease, which is river blindness. So onchocerciasis is a disease of Central Africa predominantly. There are roughly 20 million people who are infected with these worms at this point, and at least 10 times more who are at risk of infection because they live near the free-flowing streams, which is where these biting flies tend to predominate. The second companion disease that we'll refer to is lymphatic filariasis, or LF. In this case, worms of the genus Brugia or Wuchereria are typically spread by mosquito bites. As these worms get into the host, they then migrate to the lymphatic system. As they grow and develop, become larger, they eventually block lymph flow and that can lead to a symptom called lymphedema. Lymphedema can occur in the limbs. When it does, it creates what we call elephantiasis, and men can get a genital form of lymphedema, which is known as hydroxyls. Lymphatic filariasis is a more widespread disease. You can see that it occurs pretty much across the tropical world. There are over 100 million people who are infected with these worms, and about a third of those actually suffer from frank disease. But beyond that, there are close to 1 billion people who are either infected who are at risk of infection. So both of these diseases, I should point out, while they are not immediately life-threatening, as you might suspect, they have a tremendous impact on the quality of life for the patients and also on their ability to be productive members of society. And so it's a severe situation. The current standard of care for treatment of these filarial diseases is treatment with ivermectin. And that's typically given in the course of what's called a mass drug administration, where a community or a region is treated, all the patients in the region are treated regardless of their symptoms. So many of you may know that the discoverers of ivermectin actually received the 2015 Nobel Prize for Medicine for their work. And in fact, this drug has really transformed the way we manage this disease. That said, it is far from a perfect agent. In particular, ivermectin is what we call a macrofilaricide. It selectively kills the juvenile worms. Because it's those juvenile worms that spread the disease, ivermectin is very effective at blocking transmission in a community. But it doesn't kill the adult worms. And as a consequence of that, they then go forward and continue to make new juveniles. And so what that means in terms of overall therapy is that if you want to actually cure the patients, you need to basically give them biannual treatment with ivermectin for as long as those adult worms are alive. And that can be, they can live for a decade or longer in their hosts. You can imagine that for these patients, which often live in difficult environments, it can be a real challenge to find your way to a remote clinic on a regular basis, a couple times a year for that long a period of time. So what we feel we need moving forward is actually an agent that would kill the adult worms, what we would call a macrofilaricide. And so the development of new macrofilaricides has been the priority for new research in the filarial area moving forward. And that's how we focused our attention as well. Our program began the way a typical drug program usually begins with a screen. And in this case, I'm sorry, I'm talking ahead of my slides. The approach we took is actually which is actually quite a novel approach and was developed by our partners. It recognizes that these parasitic worms actually have parasites of their own. In fact, they live symbiotically with a bacterium known as Wolbachia. Now, we don't actually know what Wolbachia does for the worms. What we do know is that the worms really can't survive without it. And in the absence of Wolbachia, juvenile worms can't grow to adulthood. And so because of that, they can't spread the disease. Females become sterile, and so they're not able to make new juveniles, new microfilaria. And adult worms in general actually live a truncated lifespan. Adult worms don't die quickly, but rather than surviving for that decade or longer, as I previously described, they will typically die in a year or so. So that suggests this really intriguing idea that you might be able to kill worms by giving antibiotics. And that's an attractive concept because, for at least 100 years now, the pharmaceutical industry has been hard at work developing a variety of novel antibiotics that work through a variety of different mechanisms. But it turns out it's not quite that straightforward. Wolbachia is not a typical bacterium, and most of the antibiotics that have been developed to date are not successful at killing it or setting it back. One class that are the tetracycline antibiotics. And as it turns out, treatment, a clinical study performed by our colleagues has demonstrated that treatment of patients with doxycycline can actually lead to a significant reduction in their burden of disease, as indicated here by the levels of microfilaria in the circulation. You can see that over a period of months, microfilarial levels drop essentially to zero, and that's caused, first of all, by the existing microfilaria eventually dying, but then also not being replaced because the females have been sterilized through anti-Wolbachia treatment. So that's a great proof of concept for this anti-Wolbachia approach. Unfortunately, as it turns out, doxycycline is not a perfect agent to accomplish this task, and that's for a couple of reasons. The first is that doxy is contraindicated both in children and in women of childbearing age. And so while it can be used carefully in case management work, it really can't be used, for example, in the context of a mass drug administration. The other challenge with doxycycline is that to achieve the results that I'm showing you here, we need to give the drug once daily for 6 to 8 weeks. And again, reflecting on the difficult circumstances that these patients can live in, it can be a challenge for them to maintain a course of therapy that goes on for that long. But I think we've got great validation here for the idea of an anti-Wolbachia approach, and essentially this establishes the challenge for our program moving forward, which is that we need to develop anti-Wolbachia agents that are basically better, stronger, faster, safer. And so that's what we set out to try to do. Here we go. Now, as I was mentioning earlier, so our program began like most drug discovery programs begin with a high throughput screen. In this case, Rick Clark, who is an AbbVIe chemist who has a long history in the antibacterial field, was able to create a relatively small, about 130 compounds, but diverse and representative subset of the AbbVIe antibacterial collection, which was passed along to our partners at Liverpool, who had developed a high throughput, high content screen. And was then screened for its activity against Wolbachia. And that screen turned up exactly one robust hit, which was the macrocyclic antibiotic Tylosin A. Now Tylosin has a long history, like 50 years or more, of use as a veterinary antibiotic in a wide variety of species. But it's never been tested in people, and prior to our work, its activity against Wolbachia had not been discovered. But it's actually quite a good anti-Wolbachia agent. In that original high throughput screen, its IC50 value was about 30 nanomolar, which is very similar to what you get with doxycycline. And so that got us pretty excited. And so we wanted to quickly move to test whether it could be equally effective in an in vivo setting. To do this, we partnered with our friends at Bonn. They have a nice mouse model that uses yet another filarial species. This is Litomysoides, which can live within mice as a host. Mice are naturally infected with these worms, and then in this experiment, then shortly after infection, they are treated with drug or with a control. And we will, roughly 6 weeks after the drug treatment, the worms are excised and examined. And we will be looking at worm length essentially as a surrogate for worm health in this study. So what you can see is that doxycycline, as you might suspect, is effective at controlling worm growth. Interestingly, and excitingly, Tylosin is equally effective. In fact, it's equally effective when it's given at a shorter with a shorter dosing regimen. And that's valuable to us because remember that one of our goals is to develop a therapy that can be given for a significantly shorter course of treatment. So that's the good news from this study, but there's also a dark cloud which hangs over it. And that is that we only see this robust response of Tylosin when the drug is given by an intraperitoneal injection. The same amount of drug given for the same period of time, dosed orally, has essentially no impact on the worms. And we pretty quickly determined why that would be. In a companion pharmacokinetic study, you can see that the drug levels that are achieved when the drug is given IP are substantially higher than what you get when the same drug is given orally. And in fact, the overall oral bioavailability of Tylosin A is less than 2%. But there's still a lot of positives to take away from this experiment. First of all, we actually have an in vivo proof of concept from our screening lead, which essentially never happens. And we've really defined what our challenges are for a medicinal chemistry program moving forward. We basically have to prepare a molecule that behaves like Tylosin, but has a substantially improved ability to get into the body. That can absolutely be a challenge, but we felt like we had a hypothesis, at least moving forward, about how we might go forward with that. You will notice that Tylosin has 5 free hydroxyl groups. That is a lot of hydrogen bonding donor and acceptor potential to try to drag across a series of hydrophobic membranes. And so our idea was that perhaps if we could either eliminate or cover over some of these hydroxyl groups, perhaps we could improve the ability of the drug to permeate across membranes, and by doing that, perhaps then also improve the oral bioavailability. And so that's what we set out to do. Okay, so let's talk a little bit about structure activity. So Tylosin is actually a pretty good starting material for a medicinal chemistry effort. It is relatively available and surprisingly inexpensive. But as you can see, it is a complex structure with a lot of functionality in it. And so you might suspect that it's going to be a challenge to do selective modifications of this structure to make analogs. And in fact, that is true. We were fortunate, though, that we're not the first people to try to do this because of its history as a veterinary antibiotic. Other groups have tried to derivatize the compound in the past. And so there's actually a significant history of efforts made to do selective modifications. And we've been able to take advantage of that work, and we've also been able to expand on it to some degree. And as a consequence of all of that, we at the end, we have roughly 7 or 8 sites on this molecule that we're able to make selective derivatives of. That's a lot of SAR, and I am going to do an enormous disservice by essentially summarizing all of that on the single slide that I'm showing here. And by the way, I'm not even showing structures, so that's even worse. But hopefully, you'll be able to take away the key messages from all of this work. So the table is fairly complicated. Let me walk you through it. The left-hand column here shows the site at which each family of derivatives, SAR derivatives, has been prepared. And you can benchmark back to this annotated structure of Tylosin to see where those changes have been made. For example, a 2' alcohol is here, and the 4' is out here, things like that. In the middle column, you can see what the impact is of making of a family of modifications at each of these positions on the activity of the derivatives against what I would call typical gram-positive organisms. A lot of this information comes from the literature. Some of it is our own work in compounds that we've done antibiotic profiling of as well. And then finally, what the right-hand column gives you is the activity, the impacts of the same modifications on the anti-Wolbachia activity of those derivatives. And so I've summarized, and then I have taken these impacts and have stoplight coded. So a green column means that you're seeing a minimal effect, maybe a beneficial effect of the modification. Yellow means that there's some negative impact, but it's not substantial. Red means that the changes are not tolerated at all. So given that, what I'm hoping that you can take away visually from this experiment, if nothing else, are 2 key conclusions. The first, I think, which is fairly obvious, is that, again, to reemphasize something I said before, Wolbachia is not a typical bacterium. There seems to be essentially no correlation between the impact of site modifications on the gram-positives and on our target of interest. So that's intriguing. What's a little more disheartening, perhaps, is that it does seem that the Wolbachia is a much more fussy organism than are the gram-positives. So lots of changes that occur without a negative impact against gram-positives seem to be not tolerated or only poorly tolerated against Wolbachia. And so that doesn't really bode well for our SAR efforts moving forward. We're fortunate, though, that there is one notable exception to that trend, which is derivatives that occur at this 4 double prime position out here on the micro sugar. Typically, derivatives at this position are well tolerated. They may even give some modest benefit in the typical gram-positives, but Wolbachia seems to love these changes. So again, here the improvements are either modest or, in some cases, enormous. So as you might suspect, from this point forward in the talk, we will be focusing our attention like a laser on this position and looking at what happens when you make derivatives at that site. Okay, so I know there are chemists in the audience, and so I should at least briefly talk about how we can make derivatives, and I'm going to focus my attention again on this 4 double prime site. So again, Tylosin has these 5 free hydroxyls. You might imagine that selective derivatization of one of those hydroxyls would be a challenge, and it can be. We're fortunate that one of the 5 hydroxyls is much more reactive than any of the others in the system, and that is the one at the 2' position that is adjacent to the basic residue on the mycaminose sugar. So this base activates the alcohol, makes it more nucleophilic, and therefore more reactive. And in fact, you can take Tylosin and just stir it with, for example, acetic anhydride, and come out very quickly with the Tylosin 2' acetate as the only product. Now, you can use this approach to make derivatives at this 2' position. You can also then use, and this is how we'll use it, you can also use this 2' acetate as a protecting group to cover over that most reactive hydroxyl and allow you to work at other positions. So moving forward, again, we're still back to 4 free hydroxyls, but now we have only one pair which sit adjacent to each other, which are vicinal to each other. And now we can take advantage of the propensity of tin to form cyclic complexes with diols. So for example, if we treat this compound with tin oxide and remove water, we can get a cyclic tin complex that looks like this. The attachment to the metal then increases the nucleophilicity of both of these alcohols in this complex. If we then treat the complex with, for example, an acylating agent, we will react, but we will react selectively on the one of those 2 free hydroxyls, which is more sterically accessible. You can see that there's a lot of steric hindrance here and somewhat less at this position. And so what that leaves us with is selectively the 4 double prime acylated derivatives. And so that's how we will make our compounds moving forward. Now, at this point, we still have the 2' ester, but we can remove that easily just by refluxing this in methanol. In the same way that basic residue has activated that alcohol for derivatization, it can now activate the solvent to do a transesterification and cleave off the acetate group. So we can make a variety of derivatives at the 4 double prime position through this approach. Okay, so we're going to start by making a bunch of 4 double prime esters. In fact, we probably made, I would say, 2 dozen of these, but here's just a representative set of those results, which summarizes what we've seen. You can see that in sort of the worst case, the derivatization of this position gives us a molecule which has a very similar EC50 to the parent Tylosin itself, roughly in the same range. More typically, we will get an improvement in potency at that position. In some cases, that improvement in potency can be pretty substantial. So you can see that the 5 carbon ester, valeryl ester, is roughly 20 times more active than the parent is. So that's great. Potency is a great thing to have. But I will remind you that that's not the reason we were making these derivatives. We were making them because we hoped that by covering over these free hydroxyls, we might be able to improve the permeability of these compounds and therefore improve their oral bioavailability. And so we'll learn that by taking them into a pharmacokinetic study. And I can show you the results of that right here. So first, the good news. The drug levels that we see, the drug exposures that we see in this study are substantially higher than what you would get if you gave Tylosin itself. In fact, if you measure the area under this drug curve, we have about an 8-fold increase in drug exposure versus if we gave the parent. Like I say, that's the good news. The bad news is that, in fact, this blue curve is not showing you the drug levels of the parent drug that we gave. No, actually, these drug levels are down here. It's essentially non-existent. What we're seeing instead is that the drug that's appearing in the circulation has had that ester group cleaved off. It's actually returned it from the 4' ester to the parent Tylosin A itself, presumably via the activity of some hepatic esterase, although we haven't unequivocally demonstrated that. So that's bad news for a couple of reasons, not the least of which is that when we cleave that ester off, we lose the potency boost that we got by putting it on. Nonetheless, I think we have seen at least preliminary validation for the idea that, yes, covering over a free hydroxyl can give us boosts in drug exposure. We just need to find a way that we can put a derivative at this position that is not going to immediately fall off metabolically. And so that's our next challenge. We actually came up with 3 different potential solutions to this problem. The first was just to stick with an ester at this position, but to just make it more bulky. I mentioned before, and you've probably noticed, that there's a lot of steric hindrance around the oxygen side of this ester linkage. Our idea was that maybe if we also put additional steric hindrance around the carbonyl side, we might be able to make this so bulky that perhaps our esterase enzyme, whatever it might happen to be, might not be able to get in there and to cleave the ester linkage. And so, of course, this only helps us if these bulky esters are active. And we were fortunate enough to find that when we made, for example, the pivalate ester, in fact, it retains good activity. In fact, it's even more potent, again, than the parent is, about a 5-fold improvement in potency over Tylosin A itself. So that's one way we might be able to solve the problem. A second possibility is that maybe we can make something that looks like an ester, but isn't. And our thought turned quickly to the idea of making carbamates at this position. It's a little harder chemically to put a carbamate at this position, but it can be done. And again, the question is this group going to be tolerated? And we found that even the very first of the carbamates we made at this position, the diethyl carbamate, was actually quite active, as active as that pivalate ester, or sorry, that is the valerate ester that I showed you earlier in the presentation. So about a 1 nanomolar compound, roughly 20-fold more potent than the parent. So it's good. We're good from that perspective. And then finally, there is really no reason for us to suspect that a carbonyl group is even required. Now, putting something other than a carbonyl derivative at this position does create a substantial chemistry challenge. And in fact, we learned that we're basically only able to react this site with very reactive electrophiles, for example, benzyl halides or methyl iodide. The good news is it doesn't matter that we're limited at that position because, in fact, these benzyl derivatives are very active and looks like they will be really well tolerated at this site. So great news. We have 3 opportunities to test our hypothesis. But remember, again, that the goal is not to make these potent analogs, but rather to find out whether these derivatives are going to, number 1, improve the absorption like we saw previously, and then number 2, whether they're going to be stable to metabolism. And once again, that will involve taking a representative from each of these series into a pharmacokinetic study. Okay, so let's start with the bulky ester, the pivalate ester. And right away, we've got some good news from this approach. So the first piece of exciting information is that, unlike in the previous PK study I showed you, now this blue curve that I'm showing you is the parent. In fact, there is no evidence for cleavage of this ester linkage in this pharmacokinetic study. Next piece of good news is that drug exposures have gone up substantially. In fact, the area under the drug curve, when it's compared to Tylosin itself, has increased by over 10-fold. Now, remember, of course, that drug exposure really only matters in the context of drug potency. It doesn't help you to increase your drug exposure if you've lost your activity in the course of doing that. But the good news for us is that that argument actually works in our favor. Not only has this modification improved our drug exposure, but remember, it also improved the drug potency by about 5-fold. So if we think of these two parameters in context together and essentially look at what we call a potency-weighted area under the drug curve, you can see that this new molecule is now more than 50 times superior to Tylosin itself in a parameter that we believe should be relatively predictive of how the drug would behave in an in vivo disease model, for example. So that's great. We've already made great progress. What about the carbamate? The carbamates are also stable. Again, the curve you see here is the parent. In this case, we are getting a little bit of cleavage. That's the curve that you see right here, but that amounts to about 1% of the total, and so it's not really something that we're very concerned about. The drug exposures in this experiment go up again. They don't go up quite as much as we saw with the pivalate ester, but they're still, roughly 7-fold better than our starting point, Tylosin A. But remember again that in this case we got a very nice potency boost by making this, putting the substituent here. So we've got about a 20-fold improvement in potency. We've got roughly a 7-fold improvement in exposure. When you think of those two parameters together in the context of a potency-weighted AUC, this new compound, this new carbamate, is about 150 times better than Tylosin in terms of its overall profile. Finally, we can talk about the benzyl ether. So again, it is stable. You might not be surprised that it's not a substrate for an esterase since it doesn't have the carbonyl linkage anymore, but it does seem to be overall quite stable. Drug exposures improve versus the parent. They don't improve as much as we might hope. There's about a 3-fold increase in AUC, not as much as we got with either the carbamate or with the ester. However, on the flip side, this benzyl ether is a ridiculously active compound. The EC50 in the anti-Wolbachia assay is about 20 picomolar, so more than 1,000 times better than where we started, just for this one simple modification. And so again, when you factor these two together, you see that the potency-weighted AUC is more than 4,000 times better than where we started out with Tylosin A. And so despite the fact that this is not the best PK profile of the derivatives we made, it does seem that the benzyl ether gives us the best combination of properties. And so it's this compound which we eventually settled on to move forward into further studies. And it'll be referred to in the future as ABBV or AbbVie 4083. Okay, so based on these improvements in weighted exposures that I'm showing you, you would certainly expect that these compounds should perform well in an in vivo model of worm disease, and that has proven to be the case. I'm just going to give you one example from a number of studies that have been done to characterize this molecule. So in this case, the worms that we're looking at are Brucia malayi. They are one of the worms that are responsible for causing LF, and they can be established actually in GERDs by intraperitoneal injection of an early stage larval form of the worm. So after the GERDs are infected, the worms are allowed to grow, eventually to grow to adulthood, and that takes about 4 months. And eventually these worms actually reach sexual maturity and they start producing microfilaria of their own. During that time, as you might anticipate, the biomass of Wolbachia increases in lockstep with the biomass of the worms themselves and in exponential form. So we eventually get a substantial amount of Wolbachia in the worms as well. Once the worms are mature, we can then begin drug treatment. In a typical study, we'll have a vehicle group, we'll have our positive control, which is doxycycline, and then we might have one or more groups of the AbbVie compound. Each of these drugs can be given at multiple various doses and also for various time intervals. At the end of all the drug dosing, we'll wait a few more weeks. for a drug to wash out and for the drug to do its work. At that point, we can extract the worms. We can look at what's happening to Wolbachia in the female worms, and we can measure microfilaria in the peritoneal space as an indication, for example, of female worm fertility. And so those are the parameters that we'll be focusing on as we move forward. Okay, so how does the drug work, and how does it compare to doxy? I'll show you 3 results from that particular study. We'll start with Wolbachia. So again, we have 3 groups. We have our control group, our doxycycline group, and the AbbVie group. You can see that, as expected, doxycycline does cause a substantial reduction, about a 2 log reduction in the amount of Wolbachia in the female worms. The AbbVie compound seems to be even better. It's actually close to a 3 log decrease in worm load, and that is despite the fact that the AbbVie compound is given at a lower dose and for a shorter period of time than doxycycline. So it seems that it's behaving as the better agent in this study. Now, we've had discussions around these results, and it might be argued that some of the numbers we're seeing are even overkill. Our worm experts, based on some clinical studies that they've done, have hypothesized that if you can drop Wolbachia loads by 90% or more, so by 1 log, that is actually sufficient to commit the worms to death. And so then the question is, when you look at the study, which gives a holistic measurement of Wolbachia loads, what happens if we then go back and re-examine the study based on what's happening with each individual worm? How many of the worms in the study have been committed to death based on the standard of having more than a 90% reduction in their own personal Wolbachia load? And that's actually shown on this next slide here. So again, none of the control worms meet that criteria. They're all quite happy. Doxycycline is able to deplete Wolbachia loads by 90% or more in the majority of these worms, but only about 70% of them. So that means that typically there would be 30% or more which would survive and perhaps even thrive. The AbbVie compound is again better. 95% of the worms meet this target. In fact, only one worm in the study failed to have at least a 90% depletion of Wolbachia. So again, it seems like from the perspective of what's happening to the individual worms, we might be doing somewhat better. And then finally, well, let's look at what's happening to microfilaria. Again, this is going to be a surrogate measurement for whether the female worms are still fertile or not. And you might expect that the microfilarial loads would track with worm fertility and that would track with the Wolbachia levels, and you can see that they do. Doxycycline gives a nice reduction in the levels of microfilaria in the peritoneal cavities of these mice. Again, the AbbVie compound is able to drop those levels still further. So all suggesting that in a variety of ways of looking at this, it seems that the AbbVie compound is able to outperform doxy, even given again at lower doses and for shorter periods of time. And so it seems to be achieving that goal that I set out earlier of being better, stronger, faster, which is very attractive for us. Okay, so we can take the results of that study and a wide variety of other studies that have been done in multiple models of filarial disease, combine them with pharmacokinetic data in these various species to begin to make predictions of what sorts of drug levels we're going to need to achieve in order to be successful at treating filarial disease in man. And so we can make those predictions based on the maximum concentrations we expect to see in those human dosing studies, and also based on the area under the drug curve. And we can use these predictions, human drug exposures, human therapeutic drug exposures, to compare with a variety of assessments we've done of the safety of the compound, both in vitro assessments and also in vivo studies done in course of toxicologic studies. What we see when we compare these drug levels with the drug levels achieved in these other various studies is that we appear to have, a safety window that at least suggests that it might make sense for us or be reasonable for us to take these compounds forward into people and expect to see efficacy at doses that could be safe. And so that has inspired us to move the compound forward into clinical trials. Now, before I talk about that, I do want to show you the results of 2 additional pharmacologic studies that have been done by Mark Hübner's group in Bonn, which do relate to the way that a compound with the profile like we're showing with 4083 might be used therapeutically. The first of these studies explores the ability of our drug to combine with another anti-worm agent, albendazole. Now, albendazole is a first-line therapy for a variety of worm diseases, but it doesn't have any effect on Wolbachia, and so it's working via a different mechanism. The question is, will those drugs then combine successfully? And to do that study, results are shown here. One way we can do this is we can begin with a dose of the 4083, the AbbVIe drug, which is not effective. And so you can see that at a dose of 10 milligrams per kilo given for 14 days, this is not sufficient to give us a reduction of Wolbachia versus the control. We give albendazole at a similar dose for a similar period of time. Again, as I mentioned, it's not an anti-Wolbachia agent, and you would not expect it to have an effect on the Wolbachia levels here. But interestingly, when you combine those 2 drugs, you see a remarkable, robust effect to reduce Wolbachia in the worms in this study. And so it does appear that these 2 drugs can act synergistically to get an enhanced effect. This could be valuable if, for example, we felt that it was important to dial back the doses of these compounds, for example, to avoid some safety signal if that was necessary. The other way we can explore the combination therapies is by rather than looking at dose, to look at time. And that's shown now in this next panel. Another slice from that same study. Again, we're going to use a dose of ABB483, which is not effective. We're giving it at 20 mg per kg per day, a slightly higher dose, but now only for 7 days. And you can see again that there's no impact on Wolbachia levels. Similarly with albendazole, as we suspect. But once again, even at this shorter dosing interval, when these drugs are combined, we can see a substantial reduction in the Wolbachia load in this study, suggesting that perhaps combinations can be used as a way to shorten the treatment interval. Again, just to remind you that that's one critical goal, that we hope to establish superiority versus doxycycline therapy. So that's one study, and again, it gave us hope for how this drug might be used moving forward. The other one, which I think is really interesting, examines the effect of how the drug might be used in the real world. So this is looking at the effect of missed doses. So in the control group, they are dosed with vehicle for 14 days. The good mouse cohort, the cohort that follows its dosing regimen, receives a dose of 25 mg once a day for 25 mg per kg once a day. And every day it takes the drug, these mice take the drug just like they're supposed to. The bad mice, or the perhaps real world mice, do pretty well. They take the drug for a while, then they skip a dose, then they take a dose, then they skip a dose. Then they remember for a while and they stay on drug, and they forget again, and they take a dose and they forget, one more time. So the question is, what happens with a regimen like this? What happens when you miss doses? And the good news from this study is that as long as these mice eventually get all of the doses of drug, they catch up at the end, as you can see here. The effects on the Wolbachia are just as good in the discontinuous group as they are in the group that stayed on drug for full time. And so that does at least imply that we might be able to use this drug in a real world setting, and if the patients behave as real world patients do, we might still be able to see a beneficial effect. Okay, so where are we with this drug? We have recently completed what we are called phase 1 studies. So this is the first set of clinical studies in people. It's done in healthy volunteers. And remember, the goal of a phase 1 study is to explore what the pharmacokinetics of the drug look like in man. Are they what we had predicted based on our preclinical pharmacokinetic studies, and also to look at safety. And so based on the phase 1 study, we looked at, first of all, at the effects of single doses and eventually at multiple dose protocols. So for drug given for a week and even for up to 2 weeks. We were able to establish a maximum tolerated dose. That is the goal of the phase 1 study. And we were able to determine what happens if you exceed that maximum tolerated dose. And again, the good news from this part of the study is that the first adverse events that we observe when the drug levels get too high are the ones that were predicted from our original toxicology studies in animals. They are readily monitorable, and the good news is that they are readily reversible as soon as the patient either completes the course of therapy or just goes off of drug. And then the final bit of exciting news here is that the drug exposures that we got with these tolerated doses appear to be sufficient, if the models that I showed you previously are accurate, appear to be sufficient to predict that those doses would be efficacious in lowering Wolbachia levels and then hopefully clearing worms in those patients. So that leaves us with the next critical experiment to do, which is where we actually do get into those patients. The design of the phase 2 study, which is the first to explore that question, is underway. As you might suspect, those studies will take place in Africa. And at this point, this is where DNDi will really play a critical role. They have developed a clinical infrastructure in Africa, and so they will take the lead on the clinical program with support from the AbbVIe clinical team as well, who has had at this point more experience with the drug itself. Okay, so I think that's where I'll leave you at this point. I want to thank you for your patience, and we'll be happy to take any questions. I know Carolyn will hopefully be monitoring how questions will work out, but I'll take them either online or if you can submit them, she will be happy to ask them as well. So thanks very much for your attention.
Thanks, Tom. It was a great talk. Lovely to hear a story all the way through like that. We do have a question from Joey, who's typed his question into the chat box. So if anybody else wants to do the same whilst I'm asking this one, please go ahead. So Joey says, in the combination study with albendazole, the dose of 4083 was doubled, whilst the albendazole dose was left at 10 mg per kg. What was the purpose of doubling the dose of 4083 and reducing the time during the trial?
Right. So it's a great question. The goal of the study was to reduce the time. We know from a variety of pharmacology studies that we've done, with this drug, that when you reduce when you reduce the time that you give the drug, it takes substantially more drug to have any significant effect. And unfortunately, that's not a linear ratio. So doubling the dose while having the time does not balance out. It's harder to achieve a result when you're at a shorter dosing interval. So we chose a dose that at 7 days of dosing did not give an effect, but that was not far below doses that might eventually cause some impact on Wolbachia levels at 7 days of dosing. So we're trying to get a dose that is not effective, but is not so far from being effective that it can't be rescued. Hopefully that wasn't the clearest answer. Hopefully you got the point out of it.
Says thank you. So I have a quick question. I was very interested in the aldehyde on your molecule. So have you got any evidence that you're getting covalent? It's clearly very important to activity because the activity went down hugely when you blocked it. So do you think there's any kind of covalent interaction going on there?
Yeah, that's a great question. We have no evidence for that. It's an interesting molecule. You highlighted the aldehyde. There's also a dienone in that molecule. So we have two pieces of chemical space there that one might qualify as reactive. And perhaps if you fed tylosin into a red flag algorithm, it would turn on all sorts of red flags because it's got several electrophiles. There's no evidence to suggest that either of those is a reactive functionality, and so presumably that's because with a molecule as big as this, those may be tucked into the interior of the molecule and protected from reactivity. But yeah, it's something we wondered about. So I can tell you that the aldehyde is chemically reactive. In the work that was originally done to develop antibiotics, several teams have made, for example, imines or hydrozones, out of that aldehyde. So it reacts chemically like you should, but it doesn't seem to in biological space. Great question.
Okay, and we have a question from Mark who says, what do you think underlies the very divergent SAR versus the typical gram-negative, I think the gram-positive bacteria and Wolbachia?
Gram-positive.
Yeah,
exactly. Yeah. So I'm not a bacteriologist for sure, but apparently the Wolbachia is neither a gram-positive nor a gram-negative, which is a fairly unusual phenotype. It has its own coat, which is really distinct from most other bacteria. So presumably that is part of what makes it different. Yeah, exactly.
We have a comment from Dale who says there is precedence from the crystal structure of tylosin to a bacterial ribosome of covalent interaction of the aldehyde with ribosomal RNA. Thanks, Dale.
Great. There we go. Okay.
There you go. Takushi says, what is known about the mechanism of action? Do you still think it's protein synthesis inhibition?
Yeah, so that's a really good question. We have not proven that, but I think we have absolutely no reason to suspect that it is not acting via binding to the 50S subunit of the ribosome, which is what it does as an antibacterial. There's been some effort on the part of our worm collaborators to try to establish that, and it's been tricky to determine it. So, but I think all the evidence implies that we're working through 50S and through protein synthesis. Yeah.
So, what are your plans for scaling up the synthesis of this compound? Is it easy
right
to
now we
produce?
have quite a bit of it. Yeah.
Yeah.
There's Dale, you'll probably remember better than I do, but we certainly have multi-kilo quantities in a drum right now, and it's quite stable. We have more than enough to take us through the clinical studies that are currently being proposed. At some point, assuming things go well, we'll eventually have to start to make more. But the route that's been developed is pretty robust and should be scalable.
Great, because of course the structure of that compound flies right against all of modern drug discovery protocols.
Absolutely, yeah.
It's too big.
Which tells you what you need to know about the details of modern drug discovery, I suppose. So,
Yep, Dale says we have enough API to complete phase 2. And Mark says, do you have any theories about synergy between your compound and albendazole?
Yeah, not specifically. I guess what I've been assuming is that while albendazole doesn't act against Wolbachia, it probably does weaken the worm, and so by doing that, it makes it more sensitive to the stress that's being put on by depletion of Wolbachia. That's kind of a hand-wavy answer, but I suspect it's something like that.
I kind of missed, what was the original idea between trying albendazole?
Yeah, so I think other anti-Wolbachia agents have been tested again, in combination with albendazole, and have shown some beneficial effect as well. So there's some sense that the mechanism can be used synergistically with that.
Okay, we probably have time for one quick question. Do any of the interns want to ask any questions? They've all gone very quiet. Oh yeah. Would this, Joey says, would this be a prime example of a molecule that goes beyond Lipinski's rule of 5?
Oh my gosh, yeah. It fails at least 2 or 3 of the rules, I'm afraid. Yeah. So.
Very definitely. But it works.
But it also there is this corollary to Lipinski's rules, which is probably as soft as the rules themselves, which says that natural products are typically exceptions to the Lipinski model. So perhaps we're getting that benefit. I think you do see with natural products that a lot of the features can form internal interactions which sort of blunt their impact. So, for example, some of those free hydroxyls may be hydrogen bonding internally, and so you're not seeing all of the consequence of their presence.
So do you think that this compound will be suitable for mass dosing?
So that is very much to be determined. Yeah, I don't think we can even speculate that on that at this point. Certainly, the path that takes you to mass drug administration passes through a lot of experience in patients before you get comfort level with giving it broadly to people who don't have the disease. So we'll need a lot more safety work before we get there.
Okay, and I think we're at the hour, so it just remains for me to say a huge thank you to Tom for a really good talk, and thanks to everybody for your questions. It's been a good session.
Thank you. Yeah, thank you all. Yeah. Cheers. Take care, everybody.
Have a good day, everybody, and I'll end the meeting here.